Welcome back to MLT 120 again. In today's lecture, we're going to be covering three different concepts. We're going to be talking about the structure of hemoglobin, and we're also going to be talking about the oxygen dissociation curve, and we're also going to be discussing the red blood cell degradation process. Let's first talk about hemoglobin structure and synthesis. We first need to familiarize ourselves with a few different terms. First, we need to know what transferrin is. Transferrin is a protein, and it's a protein that transports iron from the liver to the bone marrow, and it's used for hemoglobin synthesis, or it's used to make hemoglobin. Ferritin is also a protein. It's a non-hemoglobin iron. It's a form of iron used for storage. Okay. Porphyrins is a heme precursor substance. It's an iron-containing compound, but it's a non-protein part of hemoglobin. Our hemoglobin molecules contain proteins in the form of globin chains. And we have different types of hemoglobin, and those different types of hemoglobin molecules are made up of different globin chains or combos of chains. And some of the types of hemoglobin that we have is hemoglobin A, hemoglobin A2, and hemoglobin F. There's also a few different types of embryonic hemoglobin. These are Gower 1 and 2 and Portland. For our purposes in our lecture, we're not going to focus a whole lot on these types of hemoglobin. So let's look again at our hemoglobin structure. We saw the same image in the previous lecture, but remember our hemoglobin is located inside of our red blood cells. So this is our hemoglobin structure. So within our hemoglobin structure, we have our iron. We also have our heme group. Remember, our heme comes from our precursor molecule of porphyrins. And we have different chains that make up our hemoglobin. Hemoglobin molecule is a protein composed of 4 globin chains. Again, these are protein chains. Also, it's made up of 4 irons and 4 hemes. So you see the different colored chains represented by different colors in this image. And these little purple structures are representing the 4 different hemes and the 4 different irons. There are different types of globin chains that are determined by the sequence of amino acids. So each chain is made up of different amino acids. So we have an alpha chain, we have a beta chain, we have a gamma chain, and we have a delta chain. So now let's compare the different types of hemoglobin. What makes one hemoglobin one type versus another type? There are different types of hemoglobin molecules because they have different globin chains. Remember, the globin chains are made up of different amino acids, and amino acids make up a protein, so the globin chains are proteins. So ultimately, hemoglobin is a protein. The first type of protein that we're going to talk about and its structure is hemoglobin A. Hemoglobin A makes up about 95% of an adult's hemoglobin. It contains 2 alpha chains and 2 beta chains. So that makes up for a total of 4 chains for that hemoglobin molecule. 2 alphas, 2 betas. Now for hemoglobin A2 and hemoglobin F, that only makes up for about 5% of all adult hemoglobin. So you have a lot less of hemoglobin A2 and F in the body. So let's talk about the structure of hemoglobin A2. For hemoglobin A2, the structure contains 2 alpha chains and 2 delta chains, for a total of 4 chains. For hemoglobin F, which is also referred to as fetal hemoglobin, it contains in its structure 2 alpha chains and 2 gamma chains. Now let's talk a little bit more about hemoglobin F and those embryonic hemoglobins. In newborns, they only have hemoglobin F, but the hemoglobin F begins to convert to hemoglobin A eventually. This process can take up to about 2 years to completely switch over to hemoglobin A. But as adults, you still have a little bit of hemoglobin F in the body, because remember you have 5%, but as an adult you're mostly going to have hemoglobin A. Now for those embryonic hemoglobins, for the Gower 1 and 2 and the Portland, these are those primitive hemoglobins that you have in the embryo and fetus before you even have the hemoglobin F, before the hemoglobin F even develops. And again, we're not going to focus heavily on these, just know that those are primitive embryonic forms of hemoglobin. Okay, here is a nice chart showing you all the different types of hemoglobins and the chains that they contain. Here is your Greek letter key over here. If you're not familiar with your Greek letters, you might want to become familiar with your Greek letters, because you will be using Greek letters to represent different things in the laboratory, in particular like here for hemoglobin chains. You'll be learning about some Greek letters in the lab calculations class. The second and third column of the image is covering your fetal hemoglobin types. And again, we're not going to focus on these first 3. We're going to be more concerned with F. And our fourth and fifth columns that contain our adult hemoglobins. And you want to make sure and commit the globin chains that belong to each type of hemoglobin to memory. So remember that our A type hemoglobin has 2 alpha and 2 beta. Our A2 hemoglobin has 2 alphas and 2 deltas. And our F hemoglobin has 2 alphas and 2 gammas. So let's do a little trivia based on the information we've learned so far. Which of the following do you think is considered a storage iron? And we've got possible options of transferrin, ferritin, porphyrins, and globins. So pause the video at this time and try to answer on your own before you go to the answer on the next slide. Of course, the answer is B. B is ferritin. Ferritin is our form of storage iron. So just to review some red blood cell structure and normal ranges, we said the size of our red blood cell is about 6 to 8 microns. The shape is a biconcave dish shape. This little divot or dip inside the center of the red blood cell is called a central pallor. The normal range for red blood cells in a human is between 4 to 6 million per microliter. So that's the target range. That's what we want to have. So if you have less than that, you're considered anemic. Now let's talk about the function of hemoglobin. The primary job of hemoglobin is to transport oxygen from the lung to the cells and tissue of the body. Hemoglobin has to bind to oxygen in order to do this. So we need a mechanism for hemoglobin to give up the oxygen that it's carrying to the tissues, and we also need a mechanism for it to bind to the O2 in the lungs. We have a term for this. We call it hemoglobin's affinity for O2. You can kind of think of affinity as hemoglobin's love for oxygen. So in order for oxygen to be given up to the tissues, hemoglobin's affinity must decrease, or hemoglobin's love for that O2 must decrease. To pick up oxygen from the lungs, hemoglobin's affinity must increase, or hemoglobin's love for the oxygen must increase. So there's 3 ways that hemoglobin's affinity for oxygen can be affected. There's the 2,3-BPG molecule, there's the Bohr effect, and there's O2 tension. So we're going to talk about each one of these. First off, we're going to talk about 2,3-BPG, and BPG represents bisphosphoglyceric acid, but you don't have to memorize that, just know BPG. This is a molecule located on the red blood cell, and an increase in 2,3-BPG causes a decrease in hemoglobin's affinity or love for O2, and vice versa. So it has an inverse relationship. The Bohr effect is how pH influences hemoglobin's affinity for O2 or love for O2. A decrease in the pH causes a decrease in hemoglobin's affinity for O2. An increase in pH causes an increase in hemoglobin's oxygen affinity. So we need to know also the normal pH for blood, and that's usually somewhere between 7.35 and 7.45. So if you have a decrease, the infinity is also going to decrease. If you have an increase, the infinity is also going to increase. The last one is oxygen tension, and this is happening in the lungs. If you have an increase in your oxygen tension, it causes an increase in hemoglobin's affinity or love for oxygen. And remember, the whole purpose of these mechanisms is in order for hemoglobin to do its job of carrying oxygen around the body, whether it's picking it up in the lungs and taking it to somewhere else in the body. We need a way for hemoglobin to pick up and to release the oxygen. So that's why we either have an increase or a decrease in the love, because it needs to either pick it up or release it where it needs to go. The next concept we need to talk about is the oxygen dissociation curve. So when we run our samples on an automatic analyzer, we map out on a chart a curve or our results on a chart or a graph. So a dissociation means that the hemoglobin molecule holds on to the O2 or it lets go of the O2. So we have a normal curve where the body is doing what it normally should, it's functioning properly, but we also have something called a shift to the right and a shift to the left. If we have something called a shift to the right, it's where the hemoglobin has less oxygen affinity. And if you will, go to figure 7.7 on page 98 in your textbook, and you'll be able to see the graph to be able to visualize a little bit easier what a normal versus right versus left shift looks like. But if you have what we refer to as a shift to the right, again, your hemoglobin has less affinity for your oxygen or less love for your oxygen, and it can be caused by different problems with the patient. It can be caused by severe anemia, so that would be a low red blood cell count. It can be caused by decreased pH or acidosis, and acidosis is a buildup of acid in the blood. It could be caused by increased 2,3-BPG, which will increase the response to hypoxia conditions, or it could be caused from something like a fever. Now that's your right shift. Now for your left shift, the hemoglobin has a higher affinity for your oxygen. It has more love, so it wants to hold on to that oxygen. And some patients might have some conditions or problems that cause this. And some of those conditions are a decrease in the 2,3-BPG, an increase in the pH or alkalosis, which is where there's excess base or an alkaline substance in the body, or a decrease in the body temperature. So I strongly encourage you to look at the figure on page 98 so you can see the curve and visually see the difference between a left shift versus a right shift. Let's do another trivia question since we just learned about the oxygen dissociation curve. Which of the following can shift the hemoglobin dissociation curve to the right? Is it A, an increase in the 2,3-BPG, B, alkalosis, C, hypoxia, or D, all the above? And you can pause the video at this time before we go on to the next slide and see the answer. The answer is D, all the above. So all of these, an increase in the 2,3-BPG, alkalosis, and hypoxia can all cause a shift to the right in the oxygen dissociation curve. The last concept we're going to cover on this video lecture is red blood cell degradation. There are several steps that go along with red blood cell degradation. The first thing that we need to understand is apoptosis. This is a programmed cell death. And again, this occurs around 120 days because our red blood cell lifespan is about 120 days plus or minus 10 days. Your spleen is the organ that starts to remove aged or abnormal red blood cells. And it starts to remove them by doing something called a pitting process to the red blood cells. The liver stores iron, Fe is representing iron, as part of the hemoglobin molecule. Then we have something called biliverdin, and I always think of green when you hear the word biliverdin. And biliverdin is a bile pigment, and it's a product of heme catabolism. Catabolism just means the breakdown of a molecule. Then we have two different types of bilirubin. We have unconjugated bilirubin and we have conjugated bilirubin. Firstly, we have unconjugated bilirubin, and this is a yellowish pigment that is made during the normal breakdown of red blood cells. For the conjugated bilirubin, it becomes conjugated in the liver by diglutaric acid to become water soluble. So that's important to remember, to become water soluble. Bilirubin was carried to the liver by a protein called albumin. Conjugated bilirubin then leaves the liver via the common bile duct. And we're going to look at a diagram of all these different steps here in just a minute. Next, in the red blood cell degradation process, we're in the small intestine. The bilirubin is being broken down in the small intestine by the normal flora or the normal bacteria. And there's a byproduct of this breakdown of bilirubin called urobilinogen. Next, we have urobilin. Urobilin is a chemical primarily responsible for the yellow color in urine. But you get urobilin by urobilinogen being exposed to air, and it is oxidized. That's how you end up with urobilin. And just to note, the majority of urobilin is excreted in your feces, actually. However, a small amount is absorbed back into the bloodstream, filtered by your kidneys, and then excreted in your urine. Now, let's look at this chart to kind of map out the information that we've just learned on these two previous slides about red blood cell degradation. Well, what's the first thing we learned? We learned that red blood cells die off about 120 days. So they go to the spleen. When they die off, the spleen's going to take them out by this pitting process. So this little kind of C-shaped organ here is representing the spleen, and these little red discs in here is representing our red blood cells, and the X is representing the red blood cells being taken out by the spleen. And And once the red blood cells are taken out by the spleen, all those products that are inside the spleen can be taken to different parts of the body. Remember the different components of the red blood cells? You have iron, you have those globin chains, and you have heme. Those products have to go somewhere. So that's what happens in this degradation process. The heme can go down and be ferritin to be stored. The globin chains are proteins, so they can be broken down into amino acids. But the heme is going to be converted to our biliverdin. And then our biliverdin is going to be taken down farther into our blood circulation to bilirubin. And that's going to be unconjugated free bilirubin. Okay? And then it's going to be taken to the liver by the protein albumin, where it's going to become conjugated and become water soluble. From there, the conjugated bilirubin is going to leave the liver via the common bile duct, and guess where it's going to go? It's going to go to the small intestine, where the normal flora of the bacteria in the small intestine is going to break it down to your bilinogen, and then it's going to be turned into your bilin, and then it can be either reabsorbed or excreted from the body. So let's do a trivia question on the blood cell degradation information that we just learned. Which of the following is responsible for the pitting process of the red blood cells? Is it A, liver, B, spleen, C, kidney, or D, lymph nodes? Pause the video at this time and try to answer on your own before you go to the next slide to find the answer. Of course, the answer is B, the spleen. The spleen starts the pitting process and starts the process of cell degradation. Sure. This concludes this lecture material. If you have any questions or are having a hard time understanding any of the concepts, please feel free to reach out to me through email, come see me during office hours, or see me before or after lab time.